Okay. Well, it's a great pleasure being here. Let me find my microphone.
Switch on top. I think.
Okay. It's a great pleasure being here. I want to thank Drs. Ray and Roy for inviting me, and thank you all for your attendance. This is my first trip to India, and it's such a pleasure being here in your beautiful country and having the opportunity to speak with you, to meet with you both in this formal sense, but also I hope to be able to chat with faculty as well as students personally as the school and the workshop progress. So I work at Drexel University in Philadelphia, Pennsylvania, in the United States. Prior to that, I was at the University of Wisconsin. And prior to that, I was a graduate student and a postdoc, taking the same sort of route that many of you are taking along the way. And throughout all this time, I've had really a very strong interest in microtubules and how they become organized. And when I stumbled upon the neuron as initially what I thought as a model system for helping to understand microtubule organization in general, I was thrilled. And then more and more I became appreciative of the neuron, not just as a model system, but really as the cell type whose interests really harbored the greatest fascination for me moving forward. So what I want to do today is I want to launch from the point that one of Dr. Hollenbeck's points of annoyance or irritation, and that's the what now has become rather textbook in terms of the organization of microtubules and neurons. And in particular, I want to share with you a few details about that toward the discovery of those microtubule patterns that define axons and dendrites. And I'll just give you a little taste of what we know about that today and kind of from a lecture point of view here for the school. And this will launch into the 3 different lectures I'll give at the workshop. So for the students here, what year are you in? Are you first year, second year, third year?
It's a mix,
kind of a mixture
from master's students to the final year PhD.
So you're all at somewhat different levels. Well, over at Drexel University, I'm director of the graduate program in neuroscience. And also I give lectures, what we call our core curriculum. So I give lectures, first of all, on microtubules and microtubule organization in general across cell types. And then what I also do, then zeroing in on the neuroscience programs, I talk about microtubules more specifically in neurons. So I think what I'm going to do is I'm going to get started at a very basic level. And to many of you, this may be a little too basic, particularly on those sophisticated questions that were given to Dr. Hollenbeck. But to many of you, it may be useful to start at the beginning and think in a very basic sense. So first of all, we come across the idea of just cytoskeleton, even before microtubule specifically, talking about cytoskeleton. And what is the cytoskeleton? The cytoskeleton, exactly as it implies, cyto is cell, skeleton, I think you know in terms of the human body, the skeleton is the muscles and the bones that really do 2 different things for the body. One is to give it shape, and the other is to provide for movements of the body. And those 2 functions, shape sustaining, shape providing, and then also movement, are really the 2 functions that the cytoskeleton provides for living cells. So architecture and transport, these are the 2 functions that the cytoskeleton serves for cells. If not for the cytoskeleton, if not for some sort of struts or 2x4s, a cell would just be a formless bag. It wouldn't even really be a sphere. It would be just a formless bag of cytoplasm. So cells, as you know, take on various sorts of shapes. Some cell shapes are simple, but others, in particular the neuron, are very, very complex. Axons, dendrites, growth cones. So in the very simplest of cell types, maybe shape is not such a big issue, and nor is transport. In a very simple sort of cell, diffusion is sufficient. There are no membrane-bound organelles. Diffusion can occur over short distances, but short distances are all that are really required. As cells become more complex, as evolution ensued and cells became more complex, then more sophisticated mechanisms had to come into play in order to move proteins around. Once membrane organelles that really were too big to diffuse, once they came into being, then complex systems, energy-dependent systems of transport had to come into play. So not only is the requirement of energy to really move things around important, but also directionality. Now, when cells got more sophisticated, there needs to be some sort of organization, there needs to be sorting, directionality. Say, for example, you can imagine something endocytosed at the cell membrane, that you might want to have directionally transported towards cell center. By contrast, something destined for exocytosis, you'd want to go in the other direction. Certain substances you may want to move into dendrites versus axons in the polarized epithelial cells that Dr. Hollenbeck mentioned. There are these two surfaces, apical and basolateral, and different sorts of proteins are sorted to one of these surfaces versus the other. So these two functions are really important, particularly in complex cells, architecture and transport. And these are what the cytoskeletal elements do. And there are really three types of major cytoskeletal elements. There are others, but these are the three major types. There are microtubules, actin filaments, and intermediate filaments. And they vary in many different ways, but they also have much in common. They're all three polymers. As you know, a polymer is simply a repeating subunit over and over, like perhaps a styrofoam cup that you might drink from, or polyester clothing. It's simply a repeat of these subunits. So with microtubules, it's a repeat of tubulin. With actin filaments, it's a repeat of actin. And then intermediate filaments vary more. So depending on whether you're talking about a neuron with its neurofilaments, so you might be talking about vimentin or keratin, then the subunits differ. Actin and tubulin are really very highly conserved. And it just goes to show how incredibly important they are. For example, you could isolate tubulin from various organisms, say from a frog, and you could micro-inject the tubulin into a human cell, and it would co-assemble with the human tubulin just fine. And that just goes to show really how conserved tubulin really is. And the same is true of actin. And this high degree of conservation just goes to show how incredibly important they are. If there were major sorts of mutations that alter these proteins in significant ways, it would be lethal. You wouldn't really even get past the first mitotic division because these cytoskeletal elements are so important for cell division. Microtubules are the spindle fibers of the mitotic spindle. Then actin forms the cleavage furrow for cytokinesis. So they're very, very important, really even for the most fundamental processes in cell biology. Intermediate filaments, without showing too much disrespect, are less important in these most fundamental ways. And that's why they can vary from cell to cell. They have more specific duties to perform for different cell types. For that reason, though, oftentimes intermediate filaments become very important for diseases, because if there's a mutation to an intermediate filament protein, it's not going to be lethal for the organism, so the organism will survive, but it'll have some sort of illness, some sort of sickness as caused by that mutation to the intermediate filament protein. The other reason why intermediate filaments are rather interesting is because they are cell type specific. Only neurons have neurofilament proteins. So quite often, even if you're not a cytoskeletal researcher, biologists will use antibodies, say, to neurofilament protein to identify neurons, or other sorts of antibodies to other intermediate filament proteins to mark cell type. So all three of these are polymers. All three of them have interesting functions to perform. The other thing about microtubules and actin filaments that are interesting and in common is they're both polar. They both have one end that's favored for assembly versus the other end, plus end, minus end. Whereas intermediate filaments are not polar. Actin and microtubules are highly dynamic, meaning left to their own devices, they're in constant flux with their subunit pool. Intermediate filaments are much more stable. So about 95%, for example, of certain intermediate filament types will be in the form of polymer. There's not really too much by way of dynamics. Also, microtubules and actin filaments are substrates for motor proteins that Dr. Hollenbeck introduced. So the kinesins, cytoplasmic dynein move along microtubules, and the myosin superfamily along actin filaments. So these are the three major cytoskeletal polymer systems in cells. What I'm going to be focusing on today are the microtubules. So as I mentioned, microtubules are polar. They have one end that's favored for assembly and disassembly. One end is really the business end of the microtubule, far much more of the addition of subunits and subtraction, that's the plus end, and the other end is more quiescent, and that's the minus end. This difference can be appreciated in vitro experiments, test tube sorts of experiments, but in the cell, interestingly, the plus end, minus end difference is even more accentuated. So the minus end is relatively quiescent in cells. So where the plus ends are is really important for where microtubule dynamics occur in cells. This polarity is not only manifest at the two ends of the microtubule, though. That polarity exists all along the length of this polymer. So if I had some markers here that I could click together and you could see that there's a polarity, or maybe if you play with Lego toys and you click them together, the polarity, the two ends, are clearly different, but that polarity is all along the length. And the reason why that polarity is so important is these molecular motor proteins you've been hearing about the past few days, these molecular motor proteins will read that polarity and they will walk specifically toward either the plus end of the microtubule or the minus end. So the microtubule polarity is really all important. It's all important for its assembly properties, and it's all important for its properties as the substrate for motor proteins. Now, one of the reasons why I like to talk about microtubule dynamics to a general audience when I give my lectures to the core curriculum at Drexel, I really like to do that, not because I'm trying to convince everybody to be microtubule scientists, but because there's some broad principles that apply. Now, in the days, say, prior to the middle 1980s, you saw a lot of this sort of thing. People did these biochemical studies on microtubule dynamics, and they did them on the basis of turbidity. If you look at a solution, you just hold a solution to the light. If that's a true solution, it'll be clear. So if you're looking at, say, Kool-Aid or something like that, it'll be clear. If it's not a solution, if it's more a suspension, then it'll be cloudy. So if you have a true solution of tubulin subunits, you can see through it. If you start to have microtubule assembly, then the tubulin is turning into polymers, and that's going to bend light going through. So as the turbidity of that solution, by turbidity I mean cloudiness, the cloudiness of that solution can then be quantified using a photospectrophotometer, a very simple device in the laboratory, and you can get these sorts of graphs that are indicators of microtubule assembly. And back in those days, people did a lot of this, and they learned quite a few things. So they learned that there was an initial kind of lag period that it took before assembly really took off. They learned that you have to have a certain critical concentration of tubulin in solution in order for assembly to occur. And really what's particularly interesting about that is that it turns out that the two ends of the microtubule have different critical concentrations for assembly. So the plus end has a lower critical concentration for assembly than the minus end. That means you don't need as much tubulin in solution for the plus end to assemble compared to the minus end. So a lot of things could be appreciated from this sort of early biochemistry, but there are also some mysteries that presented themselves. For example, you had to have a certain temperature, it had to be warmed up, 37 degrees. You had to have some magnesium present. But most interestingly, you had to have GTP present. And what's particularly interesting about that, you know what GTP is? It's like ATP, it's guanosine triphosphate. It doesn't yield as much energy as ATP, but a little bit of energy. So what was particularly interesting about that is that it turned out that you really don't need GTP hydrolysis to get microtubule assembly. You just need GTP. Because if folks would use a non-hydrolyzable analog of GTP, then the microtubules would assemble just fine. So you need GTP, but you don't need it to be hydrolyzed. Well, why would that be? And I remember when I was a graduate student, I had this textbook. I can't remember what it was called. It was a big, fat molecular biology textbook, and there was a chapter all about this, how you need GTP, but you don't have to have a hydrolyzed. And it totally baffled me. I could not crack what those pages meant. And maybe that's a tribute to me as a young scholar, because it turns out it was all just garbage anyway, because they didn't know what was going on. And in the middle 1980s, Tim Mitchison and Mark Kirshner really figured this out, and they presented what was called a model at the time, called dynamic instability. Now, we just call it a mechanism. It's such a sure thing, we just call it a mechanism. And the way that dynamic instability works is like this. The tubulin, the beta tubulin, must be bound to GTP. So we're talking about free tubulin in solution. It's a dimer. Alpha tubulin, beta tubulin form a dimer. And the beta tubulin in the free tubulin is always GTP, has to be GTP. Then what happens is that those GTP tubulin subunits add on the plus end of the microtubule. What's going to happen is they're going to gradually get hydrolyzed to GDP tubulin along the length of the microtubule. That just takes a little time. That's actually a stochastic process. It just happens in its own time. As it happens, there's energy stored up in the microtubule. This is another important idea to keep in mind. Microtubule assembly, disassembly, microtubule dynamics can get some work done in cells. And you can't get something for nothing. You can't get something for nothing. So that GTP hydrolysis is storing up energy. Think of it like a spring storing up energy. So if you had a microtubule that was assembled from non-hydrolyzable analog of GTP, that microtubule would assemble, but it would be unable to get any work done because you don't have that stored energy. So as long as there's what we call a GTP cap, as long as there's some GTP tubulin at that plus end, that microtubule will just keep assembling, keep assembling, keep assembling. But hydrolysis is trying to catch up. It's always trying to catch up. And sometimes, just stochastically, it'll catch up. And the GTP cap will be lost. And if the GTP cap is lost, then catastrophe. Catastrophic disassembly. That microtubule will just disassemble.
I have a question. Sir, if we are using non-hydrolyzable, as in that case also these monomers, they are combined together, they are formed the polymers. So entropy is decreasing. So if energy is not being utilized, how that process is it?
I think the process is probably very much like crystal formation. You know, it just follows the typical rules of, say, crystal forming. You know, you have sufficient tubulin in solution, and you simply get a polymer forming, but the polymer does not have any stored energy, and nor will the polymer undergo catastrophe, because you won't ever have GDP tubulin destroy the GTP cap. Yeah.
On that subject, while we're sort of analogizing, I think it's useful to think of the actin-ATP interaction and the tubulin-GTP interaction, kind of the way you think about a G protein. Right? A G protein, say a trimeric G protein, it binds GTP and it changes its conformation, and that makes it active to do something. But it's an intrinsic molecular timer, because eventually the hydrolysis activity hydrolyzes the GTP, returns to its original conformation, and shuts itself off. And I don't know if the physics is valid, but I think of the cytoskeletal subunits that way. So you have a dimer, it binds GTP, it changes its conformation a little bit, makes it highly favorable for assembly, kind of like a G protein being activated. Becomes part of the wall of the microtubule, but then after a while, like a G protein, it hydrolyzes it. Now it's able to avidly fall off, except it's buried in the wall. But put it at a free end and it'll fall off. So kind of the way a G protein ensures that current information doesn't become permanent information. I like to think of the tubulin subunit as being like the G protein in that the state in which you want to assemble that microtubule is in this microsecond. It's not permanent information because it's going to quickly become a subunit that will avidly fall off given the chance.
Sir, is it known that after GTP binding there is a conformation change in alpha beta tubule?
It's at least assumed that a conformational change. I don't know whether that's been measured by structural biologists. Do you know that?
I would think so, but I'm not remembering any specific
Three
paper.
phosphates versus two phosphates in that site ought to make a little difference.
But there's a functional difference, right?
Well, yeah, the functional difference.
That's
There
clear.
has to be a structural difference.
There would have to be, yeah.
Can't be magic. Good point.
Sanjay, were you going to say something?
I just had a question. If you lose the cap at one protofilament, does that mean that just that one protofilament can?
I don't think so. I mean, I think it's a GTP cap. I mean,
The
it's.
whole ring has to be.
I think so. Yeah. But you see, what happens though? Is that if the microtubule is short, and that catastrophe starts happening, then you're going to lose the microtubule. But if that microtubule is somewhat longer, it's kind of a competition between catastrophe and trying to reestablish the cap. And if you reestablish the cap, then it's called rescue, and then things can go on. So it may not be, maybe you've got a point, maybe it's not an all or nothing kind of thing. Maybe both are kind of working at the same time so that you can, rescue is when we have just enough of the protofilaments to, get things going again. So one of the things that I really like about telling the story, to students particularly, is that in the old days, when thinking about that cuvette and the microtubule solution getting cloudy, more turbid, people were thinking about all of the microtubules essentially doing the same thing. We're thinking about all these microtubules side by side in this test tube, assembling, getting a little longer, getting shorter. And in fact, that's not at all the case. It's not all the case because this is a stochastic process. You can have one microtubule in the population undergoing this rapid assembly, and the microtubule that's right next to it with the same concentration of tubulin subunits. Everything is the same, and that microtubule is undergoing catastrophic disassembly.
What made one end of the microtubule positive and one end other end as a negative? How we define as a positive?
One thing that I always tell my students, don't call them positive and negative. And the reason why I say don't call them positive and negative is that it brings to mind charge. And I'm not talking about charge. I'm talking about plus n being favored for assembly and minus n being less favored for assembly. And it really just comes down to the structural polarity of the tubulin subunit. The plus n has the beta sticking out, the minus n has the alpha sticking out. And just by virtue of that structural difference, the plus n is favored for subunit addition over the minus n. But it's interesting because I sometimes get on my test answers, students will say this plus and minus. I remember sitting down with a student one time in my office and saying, it's not charged, don't say positive and negative. The student kind of looked around and said, are you sure? And I said, don't get that. So dynamic instability is really one of those underlying principles that cells are using to control their microtubular rates. Another thing to keep in mind, though, the key to this mechanism is something called selective stabilization. If you have a cell type, let's say a very simple cell type, let's say it's a migratory cell type, you may want to have those microtubules firing in all directions. And then, say if that cell is to take a turn to the right, then you want to have certain microtubules in that direction selectively stabilized over the entire population so that the cell turns in that direction. And that can be accomplished through capture of the plus ends. That could be accomplished by the binding of microtubule stabilizing proteins like tau or STAB or double portin along the length of the microtubules. So selective stabilization is very important, and particularly in the nervous system, neurons are infamous for having very stable microtubules, which is true. But neurons also have regions of the microtubules toward their plus ends that are still very highly dynamic. And the dynamic properties of the microtubules and neurons are really going to be important, say, for things like growth cone turning and navigation. So microtubule dynamics, really one of the fundamentals about microtubules. And the other thing, as I mentioned briefly and as the previous speakers talked about extensively, is that molecular motor proteins like dynein here and the kinesin superfamily here, they will interact with the lattice of the microtubule, which bears that polarity, and they will walk specifically toward one end or the other. And when I use the word walk, I'm using it for a very specific reason, that the way these motor proteins work is much more akin not to roller skates, but to a walking motion. And when people walk, what defines a walk? What is a walk is that you've got one foot in front of the other. You've always got one foot on the ground. And that's the idea here, that you don't want these motor proteins to fall off the microtubule. So by having one foot engaged all the time, or most of the time, then you're enhancing what's called the processivity of the motor, how long they'll stay in the motor. Some motor proteins are more processive than others. They stay on the microtubule more than others. But this walking motion is really key to processivity. And that's what we see here. So here's the big idea. If you have molecular motor proteins that can move toward one end of the microtubule or the other, and you have these motor proteins that are able to recognize different cargo, what else do you need? All you really need now is to organize your microtubules relative to their polarity. Like in this cartoon, you put the minus ends of microtubules over there, and then these yellow guys, imagine they're interacting with dynein, they can rush over there. You put the plus ends over on the other side of the room, those blue guys are interacting with the kinesin, and they're rushing to the other side of the room. So all you really need to direct traffic in cells and to generate asymmetries of organelle distribution is to organize your microtubules into distinct patterns of polarity. So if you think about those epithelial cells that Peter brought up, and they've got their, what is it, minus ends up the apical surface, you know, then that's one of the ways. I'm not necessarily saying it's the end of the story, but that's one of the fundamentals on how that cell is going to be able to direct certain proteins, certain organelles, up toward the apical surface, and then other ones toward the basolateral surface. And I think there's no cell type in all of nature that's going to accentuate that theme more than neurons. I mean, here's a very simple, this is what you see in textbooks, and this goes also to Dr. Hollenbeck's point, that you can't just look at schematics in textbooks and appreciate everything, but it's a very simple model. You've got the centrosome in the middle there, that's the microtubule organizing center, and then out projected from the centrosome, you've got the plus ends of microtubules. So you might have something endocytosed here at the membranes, send that toward the nucleus, that'll interact with the dynein, and you've got something here that you want to send outward, interact that with kinesin, and pretty soon you're directing traffic. Well, now let's talk about neurons. I think most of you would agree with me that of any cell type in all of nature, neurons are the cell type whose morphology, whose highly asymmetric morphology, is most intimately related to the unique work that that cell type has to do. A neuron has a small cell body that might be in your brain, your spinal cord, maybe your peripheral ganglion. And that cell body, say in your spinal cord, could send out an axon that goes all the way to your toes. So you would have in a large animal, you might have an axon that's over 1 meter long. That is just a phenomenal shape for a single cell to take on. And if you think about the challenges, architecture and transport is really just profound. Let me double up on that. Not only do neurons generate an axon that can effectively grow and grow and grow and grow and grow and grow until it reaches its target tissue, neurons also generate dendrites. They do both. They do different things. And these two processes are different in many ways. The axon is designed to keep growing until it reaches its target. Dendrites, by contrast, they grow for a while. They don't get all that terribly long. Some are a little longer than others. And they just stop growing. Axons, generally 1 per neuron, talking about vertebrate neurons, generally 1 per neuron. Dendrites are multiple. Dendrites are short, stout, tapering processes. Axons are long, thin, slender processes. Dendrites branch a lot, typically. The whole idea of dendrites is they're receptive. So the axon is sending out information, dendrites are receiving information. And you can see here, they communicate through synapses. The whole idea of the dendritic arbor is that you're increasing the surface area for synapse formation. You're just creating a lot more cell membrane. I mean, they could synapse on the cell body, that would be okay. The dendritic arbor gives a lot more space for synapse formation. And then one more thing. Internally, compositionally, they're different. One of the biggest differences is that dendrites contain Golgi outposts. And even big fat dendrites might have even Golgi stacks in their proximal region. Axons don't contain Golgi. Now, what if I told you that Golgi elements move specifically toward minus ends of microtubules? They engage dying, they move toward minus ends of microtubules. That's why in most cell types, the Golgi stack is at cell center. It's crowding in on those minus ends of microtubules. Well, now, doesn't it make really perfect sense that dendrites would contain Golgi, but not axons, if I told you that dendrites contain a very different pattern of microtubule organization than axons? So, the whole idea here, collectively, is neuronal polarity. Neurons are the cell type whose polarity is most intimately related to the work that they have to do. I've talked about differences in morphology, composition, and function. Those are all collectively elements of neuronal polarity. And I introduced the idea of microtubules really being important for that. Microtubules fill up these processes. They're not attached to the centrosome. They're free. They're multiple lengths, short and long microtubules. This is just an immunofluorescence image of a cultured neuron. This is very early on in culture. You can just see it's immunostained for microtubules, very, very prominent structures. Now, let me talk about those differences in microtubule polarity orientation. This is what we found for vertebrate neurons. The axons have nearly uniform orientation, plus and distal. Almost all the microtubules are lined up with their plus ends pointing outward. Now, this was known for a while. This was known, discovered, and reported in 1981. And then a while later, what we found is that if you look at dendrites, it's different. It's different. The dendrites do have plus and distal microtubules, like axons, but they have something that axons don't have. They have minus and distal microtubules. So dendrites have a mixed orientation, non-uniform mixed orientation. It's a very different patterns of microtubule organization. And what significance might those patterns serve? Well, one of them I mentioned with regard to Golgi as an example is that certain sorts of organelles that will move toward minus sensitive microtubules, they're not going to get into the axon, whereas they're going to get into dendrites. Dendrites are going to entertain both plus end-directed organelles and minus end-directed organelles. And in fact, if you look at a histology textbook, if you look at sections of axons and dendrites, you'll see that dendrites look an awful lot like cell bodies. They have just about everything cell bodies have, except for the nucleus, of course, whereas axons, they look sort of empty. They don't really have everything. They have hardly any ribosomes. They don't have any Golgi.
Axon, we have tau, but in dendrite, there's a MAP2.
Right.
And MAP2 stabilizes microtubule. Generally, MAP2 stabilizes microtubule are very small. And tau actually is very longer microtubules.
There are differences in microtubule-associated proteins in axons versus dendrites. I don't know if that is the direct result of these polarity patterns or if that is something else. So, but that is another major difference, is that axonal microtubules will be dominated by tau and dendritic with MAP2. Scott?
One thing that should be kept in mind is that although you will very routinely see tau described as being present only in axons,
I knew you were thinking.
So is tau found in dendrites also?
Tau's in dendrites. It's just a different phosphorylation state, so it's not recognized by tau-1 antibodies. So
the
yeah.
problem is that almost everyone uses tau-1 as a marker for axons. It's a great marker. It sees a dephosphorylated epitope on tau, and it's dephosphorylated in the axon. But this phosphorylated cell body is so what happens is that although there's probably more tau, there is tau in the locations. It's just a differential.
And young people, be really careful with markers. Nature doesn't put proteins there to mark stuff for you. They're doing something. And it's very easy to get sold on the idea of a particular protein, or in this case, phosphoisochromic protein, being a marker for a particular compartment because you like the way your first 5 stains look. Very cautious.
You know, when I was younger and I'd submit papers for publication, I'd often get these reviews back from people who would say, how do you know that's an axon? How do you know that's a dendrite? Why aren't you staining for tau and MAP2? And it made me so angry because, for one thing, the morphology, just the morphology alone is better than any marker. But moreover, the thing is, if you're working in culture, the first few days anyway, tau and MAP2 are really not compartmentalized in any dramatic way. So sometimes people think they know more than they do, and sometimes, as Dr. Hollenbeck indicates, sometimes these markers are overrated. Yeah, okay, we have a bunch of...
How does the mixed neuronal polarity in dendrite help in dendritic function, and for that matter, what is the real function of dendrites?
Okay, all right, talk about that a little bit.
So another question that in, if we take pure tubulin, test tube, and add MAP2 or tau, actually if you look light scattering assay, you will not be able to distinguish tau and MAP2. But if we look at them under different microscope, you will see the MAP2 made many microtubules that are smaller in sizes, and tau are probably bigger microtubules. So from that, it's not really able to distinguish. And if you look at the effect of MAP2 and tau on dynamic instability, particularly adult tau and MAP2 are very similar. Fetal tau and MAP2 are different, but fetal tau and adult tau are also different. Now, how do you actually reconcile that MAP2 functioning in dendrite and tau in axon from the in vitro known functioning of
I mean, I think it's rather mysterious exactly what tau and MAP2 are doing. These are complex molecules with high numbers of phosphorylation sites. Clearly, they must be doing something. One of the cartoons, getting back to Peter's talk, that I particularly dislike is an infamous cartoon that shows in Alzheimer's disease that when tau comes off microtubules, they fall apart, which I don't think that microtubules will become any less stable for loss of tau because there are other stabilizing maps that are present in axons. I have a feeling that maybe what MAP2 is doing in dendrites is maybe impacting the electrical properties, because dendrites and axons are different electrically. But that's just a guess based on some old literature from Shelley Halpane and others. So I think the bottom line is it's a good example of the point that Peter brought up, that there are frontiers for exploration that seem like the most basic questions that you think, well, that's done, that's settled. In fact, it's far from all settled. So I want to get back to your question, because that's really on the direct path of my presentation.
Whether these tau proteins doesn't involve in stabilizing these microtubules inside the cell body?
I don't know, because let me tell you, you can take a test tube and you can put in a lot of tau, you can put in a lot of MAP2, you can put in even a lot of motor protein. As long as it binds up the microtubules, it will stabilize the microtubules. And does that necessarily mean that at the concentration of that protein inside the cell, that's what it's doing, is serving as a stabilizing protein? And I think that question is still unknown. I mean, you can overexpress tau, and it'll make microtubules extremely stable when you overexpress it in cells. But I'm not sure, I'm not convinced that tau is present in neurons at a concentration that it could be expected to stabilize the microtubules. And even if it is, I'm not sure if you took it away from the neuron that the microtubules would be any less stable, because they also have several other stabilizing proteins. So this is just a bit of lore in the literature that I think has not been tested sufficiently.
No, in a very low concentration of tau, actually even 1:50 equivalent is to tau.
Yeah.
Tau does stabilize microtubules. Actually, both, when I was in Les's lab, I have a couple of papers on tau stabilization, and Tony Hyman with Marcus Nardo, with all, showing that at a very low concentration of tau can stabilize dynamic instability. So that is probably the concentration range. We don't have much lower concentration in cells than the concentration that we tested, 1:100, 1:50.
Okay, well let's agree that tau is capable of stabilizing microtubules. But I can think of other studies in which tau has been deleted from axons, and when it's been deleted, say with siRNA or antibody injection, the microtubules don't seem to get any less stable. So I think tau is one contributor potentially. Do you have something to add?
Yeah, well, I mean, I think one of the things that one has to be careful about what you mean by stable, because stability that most people talk about for microtubules in vitro itself is typically just showing changes in dynamics. That doesn't necessarily mean they're more stable overall in terms of challenges. In addition, we shouldn't forget the post-translational applications. So we have a paper that's just now in the press that shows a novel post-translational modification of two levels causes extreme stabilization of axonal specific. So, and I think another thing that will happen, that at the same time, there's no question that how to alter the dynamics of dynamic instability that you talked about earlier, and that may be part of this function, but I think it's probably more important other functions, as you argue. I think I would throw out another one is that it's a scaffold for other proteins. So I'll talk a little bit about this on Monday.
Okay. So I think we need to move on to my favorite question, and that's, what is the relevance of these microtubule patterns? And I think one of the things that's important about these microtubule patterns is they fundamentally assist in directing traffic. So that's one of the things. These are two compositionally different types of processes, and having underlying differences in the microtubule polarity patterns is not going to explain everything about how they're compositionally different, but it's going to set the groundwork. When we did an experiment, maybe we can talk about it during the workshop, where we removed the -N distal microtubules from dendrites, they, by all things that we could measure, became axons. And that clues. So I think these -N distal microtubules are really part and parcel with the very definition of being a dendrite. Let me give you one more example. When the axon grows, the way the axon grows is for kinesin to engage little vesicles that go all the way to the tip. That's called the growth cone. And those vesicles will add on to the tip, and the tip will keep growing and growing and growing. There's a unidirectional vector, because those microtubules all have this +N distal orientation for the axon to keep growing. Whereas, if one of those vesicles would say move into the dendrite, it would move for a while along a +N distal microtubule. Then it might fall off, and it might engage the -N distal microtubule, go back to the cell body. These organelles would pretty soon get confused. They don't have a unidirectional vector to the tip. So instead of the dendrite getting longer and longer and longer and longer, it gets wider. It takes on a tapering morphology, and it has a limit to how long it will grow. Now, we still need to experimentally test that, but I think that's very easy to envision how it is that these different microtubule polarity patterns can really impact both organelle distribution as well as the morphology of these processes.
Sir, can we do in a test tube that by depleting the brine, we can convert all the dendrites to axons? All the cells that we can
in the test tube? You mean in a cell culture?
Something.
Yeah, we've done that sort of thing. I'm going to talk about that at the workshop, where what we've done, first of all, we've got to know the mechanism. We've got to know the mechanism that adds the -N distal microtubules to the dendrites, and I think that we do, and I'll talk about that a little later this morning, but also during the workshop. And yes, if we deplete the -N distal microtubules from dendrites, they become long, they become slender, they start taking on the organelle distribution, and even the map composition eventually of axons. So that's really, I think, perhaps the most compelling reason to think that it's those -N distal microtubules that are really part and parcel of making a dendrite. We got kind of a surprise from the Drosophila people a while back, when the Drosophila people found that dendrites contain almost uniform -N distal microtubules. So I really think that it's not perhaps the mixed orientation. What it really is that makes a dendrite is having -N distal microtubules. Question?
So are you trying to say that simply by having a mixed distribution versus uniform distribution, one can regulate identity, and maybe because the same motor can function both ways in a dendrite versus in an axon?
Yeah, that's essentially what I'm saying. I'm not saying that will solve every last mystery of neuronal polarity. There is a lot going on with neuronal polarity. What I'm saying is that by having two very different structural patterns, different groundwork in these two types of processes, that can form a foundation upon which the other several differences can be constructed. So I might have a hard time explaining how these polarity orientation patterns account for, say, the distribution of microtubule-associated proteins, like the top of that tube. I couldn't explain everything in an easy fashion, but I think they provide that initial groundwork for making these two different types of processes. And if they didn't have those two different types of processes, everything else will fall into line to make them the same.
don't you think that actin is more important in determining the growth and length of these dendrites and any orbital as compared to microtubule, because those are the first layers which will grow with the growth hormone and other structures, which will be the main contribution to the cell membrane and expansion of the membranes.
In the world of biology, sometimes it's a little hard to prioritize importance. And I know that I did with intermediate filaments, but I like to pick on intermediate filaments. I wouldn't necessarily say that microtubules or actin, either one is more important. They just have different work to do. I would say where actin becomes particularly important is in the early acquisition of polarity. Think about the cell body of the neuron as it's just beginning to form processes. The cell body is relatively spherical. It's starting to put out lamellipodia. Then some of those lamellipodia, which are actin-based structures, coalesce into several similar processes. And then one of those processes becomes the axon. Long before dendrites even appear, one of those processes starts to grow particularly long, particularly quickly. That's defined as the axon. And those early events, I think, are really particularly part and parcel with the actin cytoskeleton. It's called breaking symmetry. So maybe you're thinking of some of Carlos Doty's work on breaking symmetry. So I think that actin is particularly important then. Now, here, when you're talking about making the dendrites different from the axon, that I put higher priority on microtubules. Back here.
Is there a correlation between the position of the centrosome and where the axon forms?
Oh, that's another big controversy. What about the centrosome? What's the centrosome really doing in neurons? And if you go back decades, you will see various papers that will say the centrosome is incredibly important. Then you'll see another paper say the centrosome is nothing, it's not important. And it goes back and forth, back and forth, back and forth. Was it Carlos or someone else who 1 year it's one thing and then he himself switches to the next, back and forth, back and forth. All important, not important. All important, not important. What I think, I'll tell you what I think about the centrosome. I think that all of the microtubules in the neuron, in a sense, have to have their ancestral origin at the centrosome. Because they want to have a uniform lattice, 13 protofilaments. They display the characteristics of appropriate nucleation of the centrosome. You don't want de novo microtubule nucleation in neurons. It would be a mess. If you had microtubules popping up willy-nilly and axons growth, it would be a mess because you destroy these patterns. So you want nucleation to be controlled. So I think what goes on is that early in neuronal development, the microtubules are nucleated by the centrosome. Same for epithelial cells, by the way. They're nucleated at the centrosome, then they come off. They're broken off. We're going to talk about severing proteins if we get to it. They're broken off by severing proteins, and these microtubules will move off into axons and dendrites. They all come from the centrosome. Now, at some point, those microtubules can start breaking up into shorter pieces themselves. You might have a long microtubule breaks up into 3. So at some point, you don't really need the centrosome anymore. And there's some evidence that when neurons get older, the centrosome kind of poops out and it loses its nucleating ability. It kind of goes, the cell kind of goes in autopilot because now you can generate more microtubules by breaking up existing microtubules. So that's how I kind of think about it. But it is still some controversy. There's some types of neurons, like migratory neurons, where the microtubules, by and large, do not come off the centrosome. And they need to tug along the cell body when the neuron migrates. So migratory neurons, the centrosome is quite important.
So what organizes some of them as minus and distal and some of them as...
Well, that's what I'm going to talk about.
Okay.
A little bit today and a lot during the workshop. Sanjay?
If you look at a neuron, a generating neuron, are microtubules generally at some length, and is that fixed?
Microtubule length if you were to look at the microtubule length, you will see a whole variety. You will see some microtubules that are very long, maybe 100 microns long. You will see other microtubules that are 1 micron, 2 micron, 5 micron, 30 micron. It is a mixture of microtubule length. And that actually becomes very important for some of the other things I'll be talking about very shortly. And that really owes to these microtubule cell proteins I'll be bringing up very shortly. Yeah.
It's cytoskeleton services of identity as axon and dendrite.
Say it again?
Cytoskeleton, the only basis of identity as axon.
Is the cytoskeleton the only basis for neuronal polarity? No, I think that cells have a lot of different things going on, but cytoskeleton provides a foundation. Let's put it this way. If you didn't have cytoskeletal differences, you would not have neuronal polarity. But once you have the foundation laid by the cytoskeleton, then you have all sorts of other sorts of things that become important.
How long will it take?
Well, if people would stop asking questions, I could get moving.
No, we can take a break, tea break, and come back.
Do you guys want to take a break? Do you want to take a break or keep going?
Now we should take a break and tea will go.
The tea has a half-life.
I don't care. One way or the other. You want to take a break?
So let's take a break
Okay.
for 10 minutes and come back quickly and we'll continue. Thanks.
Sorry, the tea comes at 10:30 and this is temperature. Not half life, full life. The full life will come.
Well, they're not stop, start. I still think that it contributes to stiffness. Well, think about the cross-sectional. You might have 15 microtubules or 20 microtubules. So there's overlap. There's no region where you don't have
Other thing I wanted to ask, in case of the growth, when it's start from the center, initially the polarity.
You mean when the growth comes first starts going?
And how this transition of the mixed polarity do?
We'll talk about that. Yeah, I mean, this is good. It's your life. Okay.
Thank you.